Let's start with part 2. Suppose 3 non-linked autosomal genes ABC control coat color. For example, ABC we have and small ABC we have. These capital ABC control the dark color and this can control the light color. When we are crossing this, they are going to be forming the offspring regularly, and as per the question they have produced 640 offsprings. That means this is a tri-hybrid cross. If you know the tri-hybrid cross, we have to put there will be chances of getting 64, nearly 64 types of offsprings. Among this, how many will be similar to parents? That is the question. Among the 64, how many will be related to parents? We are considering two types of genotypes here. Let us do a small mathematical trick to identify this. When we go with 2 by 64 into 640, 64 ones, 64 tens, which is going to give 20. Hence the answer will be 20. That means again the 10 will be dominant and the other 10 will be recessive. This is one way of finding the answer. I think you may always want to research and find the exact answer. Here you can go with tri-hybrid cross and identify that as well. 112. In a population of families having 3 children each, the percentage of population of families having both boys and girls. They are saying there are certain families among which there are 3 children. Among the 3 children, they have both boys and girls. How many boys and how many girls, they did not say. Hence we can go with some assumption. In one condition, let's say we have the 2 boys and 1 girl. Here we have the possibility of 3 children, and as we know the probability always 8. How we find out the 8 here? It's simple. For example, in case of the XX, the maximum possibilities of XX in case of 1 girl child will be 2. Whereas for the 3 children, the 2 boys will be maximum is 6. 6 2 is 8. I hope I'm sounding clear for you. Among the 3 children, the XY possibility is 6 because 2 3 is 6, whereas in case of 1 child it will be maximum 2. When we sum up both, we'll find 8. In this scenario, 3 by 8, again there might be one more chance. Let's say if there are 2 girls and 1 boy, here also the possibility is same, 3 by 8. Let us try to find the LCM of both. 3 by 8 plus 3 by 8, 75%. I'm sorry, I did a little wrong here. 8 plus 8 is going to be the 8, because I can go once. And to identify the percentage, 6 by 8 into 100, we can easily go with it. 2 4s, 2 3s, 4 1s, 4 25s. 25 into 3 is nothing but 75%. Hence we can find the correct answer 75. I hope this is clear for you. Moving on to the next one. As an indicated gel image, lanes X and Y represent samples obtained from a circular plasmid. Here again we got the plasmid. Complete digestion using a restriction enzyme X or Y with different sites. Respectively, how many sites of X and Y are present in the plasmid? And here they mentioned with kilo base pairs. First of all, I want you to observe this very clearly. The first thing is the X location has 6 kb, 4 kb separately, and Y is little bit thicker. Why it is thicker? Because it has two regions with the same size. I wanted to be clear with this, then only we can understand the exact cause for it. As we know, plasmids are in circular position. Let me take 2 here with 2 colors. It will be clear somewhat. Now they are saying the X region has 2. That means, let me put some points in the color change. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Among this, they said 6 kb on one side. That means we need to halve exactly 6. 1, 2, 3, 4, 5, 6. 6 cut up here and 4 cuts. That means there are 2 for X region. Now let us do same for the Y. As we said already for Y, again let me put 10, 3, 4, 5, 6, 7, 8, 9, 10. And there are 2 locations. If you want to get 10 in the sense, there should be one split. On either side it has 5 kb and 5 kb. Hence we can find the locations. Though it has same, but it has 2 locations. So 2 for Y and 2 for X. Hence we can say the option 4 is correct. This Matthew Messels and Franklin Stahl grew E. coli during doubling time 20 minutes in a medium containing 15 NH4Cl. This is exactly NCERT question. 14 NH4Cl after 40 minutes. So let me say they have allowed experiment 1 for 20 minutes and they left it. Again in experiment 2 they have extended the time for 40 minutes. This is overview. After 40 minutes the cells are harvested, subjected to cesium chloride density gradient centrifugation. The proportion of light and hybrid DNA densities will be. We notice here, first of all, this is occurring in E. coli, which shows, or they have did this experiment to prove semi-conservative replication. The semi-conservative replication in the sense, always there will be one parental type and one, let's say there is a parental type and there will be one new strand of DNA. So as this occurs for n number of generations, if this occurs for 1000 generations also, the rule is not going to change. There will be always one new strand and one parental strand. That means you can find there will be always 50% light ones and 50% hybrid ones or dense ones. Hence the option 1 will be correct. In a population interaction between species X and species Y, which one of the following is correct? This is very easy. There is X that is benefiting, Y is not, Y is disadvantaged. Competition is not like that, so it is not correct. Where both X and Y are benefit, mutualism, this is correct. Both X and Y are disadvantaged, predation is not that, one will be advantaged. And both X and Y are disadvantaged, it is called parasitism. This is also incorrect. In these conditions one will be benefited, and here also the two organisms may not have food in competition. Next question. The protein P oligosaccharide O oligonucleotide, oligosaccharide and oligonucleotide, correct. N composed of 100 amino acids, 100 hexose residues and 100 nucleotides respectively. Which one of the following orders weighs molecular weight is correct? So they are asking you to identify molecular weight approximately and set in the order. This again a tricky one. First of all, we must know the proteins will have, as per the residues they have given, there are 100 amino acids, they have the least molecular weight, and then we can say the oligonucleotides and then the nucleotides, the proteins of 100 amino acids, they'll have high molecular weights. Then it comes to the oligonucleotides, then oligosaccharides, then nucleotides. So if you go with highest order, here the question they've asked for less than, hence proteins have less amount of weight and the nucleotides have highest amount of the molecular weight, hence option 3 is correct. An octopeptide sequence is given and we need to identify protease chemical digestion based on the results obtained. Choose the incorrect option given below. First of all, we must know the enzymes trypsin, cyanogen, bromide and chymotrypsin. What is their breaking amino acid? If you are clear with this, then we can be able to get the incorrect option here. These are same sequence. First of all, we'll see what is the cleavage site for trypsin. We already understood what is the cleavage point refers to where it can cleave. First, it is cleaving at the lysine and arginine. So cut this, the one amino acid and 3, and at tryptophan it is cutting. So this is what the first option says. Trypsin generates mixture of dimer and trimer. Here itself we can conclude the correct answer will be option 1. But we'll also see how the other options are correlating. The second option are the chymotrypsin. The chymotrypsin can be able to break down the sequence at the phenylalanine. Let me take the other color. This one will take the phenylalanine or tryptophan and tyrosine. So here tryptophan or tyrosine. The option chymotrypsin says generates the mixture of dimers and trimers. Obviously it is going to produce the dimers out of 8 or trimers. This is correct. So option B is correct. And next option C, cyanogen bromide generates hexamer and dimer. The cyanogen bromide can cut the sequence at this particular region. Let me take the other color. After methionine, it is going to cut, and here this is a dimer, and other one is hexamer, the whole sequence. Hence, we can conclude option C is also correct. And the fourth one, chymotrypsin generates a mixture of dimers and trimers. I think we have already covered this. Out of this, option A, which is incorrect as per the question, and our correct option is this. Trypsin generates a mixture of dimers and trimers, which it is failed to do when we have seen enzyme wise. Next, match the enzymes with column 1 with the respective biochemical reaction in column 2. The correct combination below. Here, the table displayed here is very important. You must know which enzyme group is involving in which process. Mostly the KVPY exams are based on enzymes specifically, so we must know all these equations. Here the correct matching will be option 3, whereas the transaminases will remove the amino acid because amines which is connected, and Q is going to be 3, addition of phosphoryl group at specific amino acid, then removal of phosphoryl group from amino acid, phosphatases, then dehydrogenases can involve in oxidation and reduction of these substances, which is the first one here. Hence we can go with option 3. Which one of the following graphs best describes the blood pressure BP? Iota to capillaries. I hope this is clear for you. Iota from heart to capillaries in the body. In order to make sure clear, iota to arteries. I am making an intermediate here. Artery. Through artery it reaches capillaries. Which graph is compromising here? If you think, we always end up with this answer, but the exact answer is 1. The main reason is we forget to think about artery. From iota to artery, there will be higher pressure. The pressure will be high, whereas from iota to capillaries, from artery to capillaries, the relatively lesser pressure is seen because they have the connectivity with both, less pressure. Hence option 1 will be correct, and this will not be correct, and there is no common pressure and there is no ups and downs. In case of abnormal conditions only you can get this. Hence we can say option 1 is correct. Next, the following pedigree describes genetic disorders in PQ, family 1 and family 2 respectively. Choose the correct statement. I hope you know the method. We should always, if they give like this, we have to use the capital A and small a, capital A and small a to identify. Here, if you see, the first generation itself got the disease. With this I can say this might be the dominant. Hence the first generation itself got it. So here the same. So between this, as these two are recessive, there is no chances all of these are recessive as well. Here, as it is heterozygous, you can see this is dominant, this is dominant, and this is not. Whereas coming to here, this is not shown in the first generation. Hence, we can say this is the recessive and non-infected dominant one. So here, all of these are dominant somehow. So I'm not filling this here, but here I'm trying to fill. So this is recessive. So there is an idea to answer this, and here if you find both P and Q are dominant traits, no, because it is not dominant. P is dominant trait and Q is recessive trait. And they are asking the correct statement. So this is the one which is matching. P is dominant and Q is recessive.